Hello students, good morning. We were learning the chapter number 5, that is Fundamental Unit of Life of class 9. Today in this session we will try to understand the cell and how does nucleus look like with the experiment of onion peel. To perform this experiment we need onion. We need an onion something like this and we have to cut that. After cutting we need to take a peel somewhat like this. Once you take the peel you have to fold it in the opposite way a little bit, so in the way that this onion should not get cracked, so that this epiderm of the onion peel will get removed very easily. Okay, so this is the onion peel. The onion peel somewhat like this. We have to remove the epiderm. Okay, this is an epiderm we had removed. After removing this epiderm, we need to put into the watch glass. This watch glass something like this, when we have to put this onion epiderm into the water for a few minutes. After that, again we have to take the epiderm from the distilled water and put it in the another watch glass which contains the dye called methylene blue or safranin. We can use iodine as well as per our availability. So here I have taken the dye methylene blue and transferred this epiderm into the methylene blue solution. So this will get stain. We keep this epiderm into the methylene blue solution. To make the solution, what we have to do, just transfer that epiderm and take some water. Along with the water, put or add 2 to 3 drops of methylene blue. Okay, that is what the methylene blue solution, and it is used for staining purpose. After 1 or 2 minutes, take this epiderm from the watch glass and we have to place it on the slide, glass slide we can say, that we use in the microscope. So this is the slide and we have kept on the slide. This is the epiderm we kept on the slide this way. Okay, now what we have to do is we have to cover this portion of epiderm by applying little bit of glycerin. You can apply, if you don't want to and want to see fresh one, you can directly watch into the microscope after putting the cover slip on this. Cover slip is a small glass portion that covers the slide or covers the desired part which we have kept. Okay, this is the portion we took, epiderm portion. We have to put the cover slip on that, and we have to observe in such kind of light microscope by putting it. Okay, if we observe this kind of sections into the microscope, we can see the structure something like this. Okay, if we draw the figure, the epiderm looks something like this. Some of the part of the cell which will appear darker as they catch the dye, as they catch more dye. Okay, so this portion over here in the middle, we can see it is more darker than the other part. And the portion and some portion will catch less dye, so they will appear lighter in color. So if we observe this structure into the microscope, it will appear according to the color of the dye. If we have taken over here methylene blue dye, so this cell wall, then the nucleus and the plasma will appear blue in color but in a different shade. Darker nucleus with a lighter cytoplasm. Okay, so when Robert Brown was doing experiment, okay, cells were invented already. Now Robert Brown was doing experiment and he has observed that there is something which is present within the cell and which is staying. It is called the nucleus. So the term nucleus was given by Robert Brown. Correct? After that, Purkinje also did some experiment. He had seen that if it is the nucleus, then what about the other part which is present, the lighter color one? And he gave the name as a cytoplasm. In this cytoplasm, there are many membrane-bound organelles present which float inside. This is the cell. If we say this is the cell, and this portion, this little dot is the nucleus. We have drawn the same little dot over here is the nucleus. This nucleus is having double layer of membrane. This is called the bilayer membrane. This is the layer 1 and this one is layer 2. So the nucleus have got cell membrane. This nuclear membrane is not continuous. It has some gap in middle or in between. Those gaps are called nuclear pores. Pores means a gap, hole. Those are the pores by which the nucleus can enter within the nucleus, or the elements which are needed that can travel from outside to inside. Okay, so this was, and the material which is present inside the nucleus is called chromatin. Understand? So this is nothing but the nucleus which looks into the cell, the dense material, the dense part which appear over here. This is the same thing that is called the nucleus. Nucleus has the bilayer membrane, or we can say the nucleus has got the nuclear membrane, and the membrane there are some gaps. They are called nuclear pores, and the material which is present inside is called the chromatin material. Chromatin material is an integral mass of thread-like structure. This is something like a thread, integral, we can say the gucha. Okay, so the integral mass appears thread-like is called a chromatin. Now, when the cell is in normal phase, or in the cell when it is not dividing, in that particular stage or phase, this material is appearing like thread, integral mass of thread. But when this cell reach as the stage of cell division, as we know, single cell will get division and one cell will be formed two different cells. So from one cell, two cell will get generated. At the time of cell division, understand? At the time of cell division, suppose this is a cell, single cell, okay, it's not dividing at this stage, so it will have the chromatin material inside them. But when they will start dividing, when they will get maturity, we can say the maturity stage, after getting mature, they will divide. At this stage, the cell will be having, so then they will divide and form two. At this stage, the cell chromatin material will form a rod-like structure, something like this. This is the chromatin material. At the stage of cell division, they will form the structure like this. Those are called chromosomes. Okay, these chromosomes we can easily see at this stage. The DNA material we can see. This is also a DNA material, but at the initial stage we cannot see. To see this chromatin material, we need to have electron microscopes, and with the help of electron microscope we can see this chromatin material. But once it gets dense, once the cell will reach at the stage of cell division, they will convert this chromatin material into the chromosomes. Chromosomes are the rod-like structure. And how it is formed? With the help of some proteins. Because basically what this chromatin material is, this chromatin material is nothing but is a DNA. Deoxyribonucleic acid or deoxyribonucleic acid. Okay, this deoxyribonucleic acid, when it's bind with some protein, they will form chromosomes. Understand? So to make the chromosome, what is there in the chromosome? Chromosome has got the DNA as well as proteins. Understand? And this is how they will form or they will make the structure something like this. We have seen this structure many times in the books, in the TV and some programs. These are the chromosomes, and chromosomes have DNA as well as some proteins. The proteins also play a very important role when the formation of this kind of formation will take place. Okay, now DNA. DNA, which is something like this in a continuous manner, and in between the DNA there are certain functional fragments of DNA. These fragments are what? Those are called the functional fragments. Functional means they perform certain activity within the cells. Okay, they do or they produce some molecules, or they will generate some chemicals which are important for the survival. Okay, so those are called the functional segments of DNA. Those are called genes. Okay, where does the genes are present? In the DNA. Okay, and what is the DNA? DNA is a kind of chromatin material which is present into the nucleus. So site of DNA is what? They are present in the cell nucleus. And what is the chromosome? They are protein and DNA. Okay. And when we can see the chromosomes at the stage of cell division. This is now the genes. What do the gene functions? What they do? Normally we have heard this gene word many times. So what these fragments will do over here? These fragments plays very important role at the time of cellular division. We can say in the unicellular organisms. Unicellular organism is what? A single cell that lives by itself, or the single cell which performs all the basic activity what the whole animal is performing. So the single cellular organisms, they with the help of the DNA, the gene which are responsible for the cell division. Okay, so cell division is what in that case? That is called the reproduction, because one cell has generated two cells. That is why it is called the reproduction. Okay, so the genes are involved. Genes are playing important role into the cellular reproduction. Second important function of the gene is they direct the chemical activity of the cell. Whatever the chemical secretion takes place, suppose we are mature now, you as you are in the 9th standard, so now the puberty stage is going on. So at this time different enzymes get secreted within our body. So these enzymes are what? They are some chemicals. Those chemical secretions depends on who? Depends on basically the gene. Gene gives a signal. They generate this different types of chemicals. Okay, there is a difference between the gene of in everyone. Okay, so there is a one individual and there is another one. The person which is taller one, and one individual is a shorter one. Suppose this gene is secreting what? Certain chemicals which are responsible for the height in the person. Okay, so this is the gene for the height, and this gene is secreting some chemicals, and if those chemicals are secreted in high amount, the person will be a taller. And what about this same gene will secrete the chemical in very less amount? If it secrete in very less amount, then the person will be shorter. So the gene will decide how the person will be at the stage of maturity. Okay, this is whatever the person, whatever the animal, birds, whoever is there, the gene are the basic thing by who can decide how the person will look like. Hope you understand about gene. Gene are present into the DNA. DNA, when they fold, they make the chromosomes, and chromosomes at the initial level there will be a chromatin material. So from chromatin material they will form the chromosomes which have the protein plus DNA. This DNA will be having the gene, and genes are responsible for secreting some chemicals, and those chemicals are responsible for different kinds of function within the cells, and they will decide how the cell will be at the maturity. Okay, hope you clear about this. Now we'll see the cytoplasm. This protoplasm word first time given by Purkinje, that is more commonly used as a cytoplasm. Cytoplasm is enclosed by cell membrane. The different types of organelles such as endoplasmic reticulum, Golgi apparatus or Golgi complex, then mitochondria, vacuoles are present in this cytoplasm. This cell membranes play important role during the function of the cell, function of any cell. This is the normal characteristics of the living organism, or we can say the living cell. All living cells, they having cell membrane. If they do not have cell membrane, then they are not the live cells. Say for an example, viruses do not have cell membrane. As we know these days, the coronavirus, this viruses does not have cell membrane. They have the surrounding body that is made up of either protein or lipids, or sometimes both. Okay, so what they are non-living objects. Viruses are non-living objects because they do not have cell membrane. Until they enter into the body of other organism, they will have and use the system of the host organisms. Host organism is what? The thing in which they penetrate, and they will utilize the things of that particular organism. That organism is called host organisms. Now they will be converted into the life forms, and they will produce their own proteins by using the host organism's machinery. Okay, so the organisms which do not have cell membrane are called non-living objects. Viruses are non-living objects until they enter into the host. Okay, now we see the difference between the prokaryotes and eukaryotes. Prokaryotes. Pro means what? That means primary. Pro means primary, and karyote means nucleus. So the cells which are having very primary kinds of nucleus, those are called prokaryotes, and the cells which are having the advanced type of nucleus, those are called the eukaryotes. That is the basic difference between two kinds of cells. So basically the cells are divided into two different categories: prokaryotic cells and eukaryotic cells. Prokaryotic cells means the cells are having very primary type of nucleus, whereas the eukaryotic cells which are having more advanced types of nucleus. So if we draw the primary or prokaryotic cell, something like this. This is the eukaryotic cell, and they will having the nucleus inside, and within the nucleus the nucleic acid, or we can say the chromatin material, is present. Okay, so the location of chromatin material in the eukaryotic cell is nucleus. The chromatin material which is over here, that is present anywhere in the cell randomly. So there will be not a fixed location for this chromatin material, and the chromatin material which is present in the prokaryotic cell, that is called nucleic acid. This is nucleic acid. Okay. So in the eukaryotic cell they are present as a chromatin material, whereas in the prokaryotic cell they are present as a nucleic acid, because they do not have cell membrane or membrane bound nucleus, you can say. They do not have the membrane bound nucleus, where the eukaryotic cells having the membrane bound nucleus. They do not have organelles, or we can say they do not have membrane bound organelles. As I have said earlier, that the eukaryotic cells which are having different types of cell organelles, such as endoplasmic reticulum, Golgi body, then mitochondria, chloroplast, vacuoles. Okay, but those all organelles are membrane bound. They are covered with the membrane. That's why it is called the membrane bound organelles. But in the prokaryotic cells, those membrane bound organelles are not present. Okay, third one, chromosome numbers. In the prokaryotic cell, there is a single chromosome, a single thread-like structure, a long structure is present. The number of chromosomes are just one, and there are more than one chromosomes present in the eukaryotic cell. If we talk about the cell size, the prokaryotic cell is about 1 to 10 micron. 1 to 10 micron bigger, or the size is 1 to 10 micrometer. Micron, or we can say the micrometer. Here the eukaryotic cell is 5 to 100 micrometer. Example over here in the prokaryotic, if we give, that is bacterial cell, whereas in the eukaryotic cell we can give any example like human, like animal, bird, mammals, whatever you can give. Okay, so this was about the difference between prokaryotic cell and eukaryotic cell.